Hi guys, I am Sanjay. Welcome to the channel Physiology at Once. The topic is erythropoiesis. It's an important 5 marker or 10 marker question that can be asked in a university exam. Erythropoiesis definition. Erythropoiesis is a process of origin, development and maturation of erythrocytes. And erythropoiesis occurs in different sites at different ages. First, in the fetal life, it occurs in 3 stages. In first 2 months of intrauterine life, it occurs in the mesenchyme of the yolk sac. This is called mesoblastic stage. And from 3rd month of intrauterine life, it occurs in liver and the spleen. This is called as hepatic stage. Last 3 months of intrauterine life, it occurs in red bone marrow and the liver. This is called as myeloid stage. And in the newborn, from the newborn till 20 years of age, it occurs in the long and all shaft bones of red bone marrow. After 20 years of age, erythropoiesis also occurs in the membranous bones. The erythropoiesis can be studied in 6 stages. In all these 6 different stages, in the erythrocytes, there will be a certain changes that is taking place. First, in the size of the RBCs, from first stage to last stage, it decreases from 25 micron to 7.2 micron, and there will be a disappearance of the nucleus, nucleoli, and next there is the appearance of hemoglobin. There is the appearance of the hemoglobin, and there will be changes in the staining properties of the cells. Before studying this, our basics is there is something called pluripotent hematopoietic stem cells in red bone marrows. There are 2 types, committed and uncommitted pluripotent hematopoietic stem cells. Committed means it will form a particular type of cells. Example, it can form RBC or it can form a WBCs. But uncommitted can form any type of cells. It can form any type of cells. It is not defined to form a particular type of cells. Committed pluripotent hematopoietic stem cells, they form colony forming blastocyte. This forms colony forming unit E. E means erythrocyte, and this later differentiates into proerythrocytes and early normoblast, intermediate, late normoblast, reticulocytes and erythrocytes. These 6 stages. The first stage, the proerythroblast, which is also called as megaloblast, in which the important events are the hemoglobin synthesis starts. And the second stage is the early normoblast, which is also called as basophilic erythroblast. In this stage, the nucleoli starts to disappearing. This is called as basophilic erythroblast because the cytoplasm are stained with blue color, because the cytoplasm is basic in nature. So it is called as basophilic erythroblast. And next stage is the intermediate normoblast. Here the important event is that hemoglobin starts appearing. It's very important. The hemoglobin appears in the intermediate normoblast. Why it called polychromatic erythroblast means, since the hemoglobin starts appearing, so the cells stains with the acidic dye also. So it is also called as polychromatic. So it stains with both, the cytoplasm stains with both acidic and also basic. So it is called as polychromatic. And late normoblast, in this stage the nucleus starts to disappearing. This is called as orthochromic. Disappearance of the nucleus is also called as pycnosis. And the 5th stage is the reticulocyte, which is called as immature RBC, where the reticulum is formed. And in the matured erythrocytes, the reticulum disappears and the cell size become biconcave in nature. Next, there are certain factors that are necessary for the erythropoiesis. There are 3 different factors. It includes general factors, maturation factors, the factors necessary for hemoglobin formation. The general factors include erythropoietin, thyroxine, hemopoietic growth factors and vitamins. Maturation factors includes vitamin B12, intrinsic factor, folic acids, and factors necessary for hemoglobin formation. It includes first class protein, amino acids, iron, copper, cobalt, nickel and vitamins. These 3 factors, let's study it one by one. First, the erythropoietin. The erythropoietin, it is also called as erythrocyte stimulating factor, which is a glycoprotein in nature with 165 amino acids. The source is peritubular capillaries of the kidney, and some minute quantities it is synthesized from the liver and the brain also. The major stimulant for the erythropoietin is the hypoxic condition. Hypoxia is a major stimulant, and actions in different stages. Productions of the proerythroblast from colony forming unit E of bone marrow, and there is development of proerythroblast into matured RBC. In different stages, erythropoietin is required, and release of the matured erythrocyte into the bloodstream. These are the 3 actions of the erythropoietin. Next is about thyroxine. Thyroxine is an hormone which accelerates the process of the erythropoiesis at many levels. Next is hemopoietic growth factors. These hemopoietic growth factors includes interleukins and the stem cell factors. They causes the proliferation of the pluripotent hematopoietic cells. These are the committed pluripotent hematopoietic stem cells. They form a particular type of cells. And next is the vitamins. Vitamins are required for the erythropoiesis because its deficiency of these vitamins, they are associated with anemia, because its deficiency causes anemia. Example, vitamin C deficiency causes anemia, scurvy is also associated with it. And vitamin B deficiency causes pellagra, along with that anemia is also associated. Vitamin D deficiency causes rickets, along with this anemia causes. Next, regulations of erythropoiesis. Erythropoiesis regulation. The mechanism controlling erythropoiesis is divided into two categories. First is functional feedback and end product feedback. Functional feedback. Functional feedback, it originates in the tissues. The major factor for this functional feedback is the oxygen requirement of the tissues. And erythropoietin is considered as a chief mediator in this feedback mechanism. And next is the end product feedback. As the name suggests, end product, the products of the red cell destruction. As the RBCs are destroyed, the products of this destruction, they control this feedback mechanism. The precise mediator of this feedback is not yet identified. If you found this video helpful, please like and share and subscribe for more videos like this. Thank you.